a very good morning to all the faculty members, participants. So welcome to day 5 of our professional development program. And here today also we have two esteemed speakers. The first one is Dr. Rajesh Chandra Mohan Das, who would be introduced formally by Professor Krishna Swami, who would be speaking on Plasmodium, as you can see. And in second session, we have Dr. Abhidat Sharma from MIT University, Haryana, who will be speaking on the session Genomics and Transcriptomics. So I request Professor Krishnaswamy to introduce our speaker, Dr. Rajesh, and then he can start his talk. Thank you.
Hello, everyone. My name is Lakshmi Narasimhan Krishnaswamy. I'm at MIT University, Haryana. It's a great pleasure and honor to introduce my friend, today's speaker, Dr. Rajesh Chandra Mohandas. Currently, he is Scientist E at Rajiv Gandhi Center for Biotechnology at Thiruvananthapuram. He's been there since 2021. Before that, from 2013 to 2021, he was at the Singapore University of Technology and Design, SUTD, where we both overlapped, right? And at that point of time, SUTD was a new place. Okay, the reason I mentioned this is even before joining SUTD, Rajesh was at SMART, the Singapore MIT Alliance for Research and Technological Innovations. He was there, and then he moved to SUTD. One thing, I mean, I can give a conventional introduction about Rajesh, that he has published these many papers and he did his master's here, PhD here. But I want to focus on one particular aspect of his career track, which is mostly students, particularly this. I'm saying this for graduate students. Mostly people think that if you do PhD in a very good lab, then your career will be good. But Rajesh's story is an example of where he did PhD from a moderate lab, right? He was the last student of a faculty who had already retired, and he had no resources and so on. He finished his PhD, and at the end of the PhD, he hardly had a good publication. But when he went for a postdoc, he worked hard and he got a science paper, right? And from then on, it never went down. He's been publishing papers after papers in highly ranked journals, and that's how he got into SMART in Singapore, and then later into SUTD. And one interesting thing about Rajesh is wherever he goes, he has to start from the scratch and set up a lab, right? And he's very good at it. He can probably write a book on how to set up labs. And currently at RCB, he's set up the what is the you're in charge of? You said the biosafety level 3, right? Yeah. So he set up a lab which has biosafety level 3 facility, and so that's really inspiring to me about Rajesh. I mean, I've taught many courses with him, had several scientific discussions with him. He's not only prolific in science, but he's quite adaptable. He diversifies his research, and he can collaborate with technologists, hardcore engineers, and anything to do with malaria, or now he's expanding to anything to do with red blood cells diseases. This aspect of his enthusiasm in doing science and to get things done really impresses me. And with that introduction, I would like to pass on the mic to Rajesh. Please take us through this today's interesting talk on dissecting red blood cell tropism of Plasmodium using multiomics approach. Thank you.
Yeah, thank you, Dr. Lakshmi Narasimhan, whom we used to call Dr. Simhan or Simhan or LK, right? So there were different varieties that people used to address him. And thank you for that nice introduction. So I hope I will not disappoint your, good remarks that you just made. And so today I will be talking about dissecting a red blood cell tropism of Plasmodium using a multi-omics approach. So right now I'm in the Rajiv Gandhi Center for Biotechnology in Trivandrum. But quite a bit of the data that I'm going to show today comes from my previous laboratory. Started at the Singapore University of Technology and Design. And one thing Simhan probably missed out was for a year in between, I was at the National University of Singapore in the Department of Microbiology. And so I'll be presenting the data that primarily came out of that engagement, and kind of will also give you an overview of what we are doing right now and where we are going to go. right? So with that, okay, so my lab, as Simhan introduced, has a focus on red cell infections, red cell diseases in general. We have a very strong commitment to work on Plasmodium, malaria. But at the same time, I'm also interested in developing new methods to look at infectious disease. So we are interested in developing new diagnostic devices, new engineering tools, as well as new drugs and the identification of drug targets using classical chemical biology and proteomics approach. So this is just to give you an overview of the different kind of activities that the lab is engaged in. But today's talk will primarily focus on Plasmodium falciparum, Plasmodium falciparum, which is the most common variety of malaria parasite, and which is also the most lethal variety. So I will introduce these two parasites, Plasmodium falciparum, which is the lethal variety primarily found in Africa, but it's also present in India and many other countries. And Plasmodium vivax, which is mostly present in the Asia and Latin America. Now, to put it in perspective, there are 97 countries and territories that are still affected by malaria. About half of the world's population, 3.2 billion people, are living in malaria endemic areas, right? So again, about 10,000 travels were affected. So now, if you look at the number right now, after COVID, it doesn't look like a huge enough number, but before the COVID, 10,000 travelers being affected by malaria appeared to be a significant number. So if you look at the problems related to malaria, there are a number of them. Drug resistance. So we have wonderful drugs that are active against malaria, including artemisinin. But if you look at their efficacy right now, we are very rapidly losing efficacy in many regions because of the drug resistance forming up. There is multi-species infection. So more than one species of malaria can infect the same human being. There is zoonosis. Malaria is considered to be a host-specific infection. So in other words, the malaria species infecting humans will not infect other organisms. But that is actually being changed. So now recently there are a number of reports showing malaria that is restricted to monkeys are infecting humans and vice versa, right? So there are a number of very, very complex situations that we are actually looking at as the country and the world is looking for malaria eradication in the next 10 years. Again, for the graduate students who are probably in this audience, there are, like I said earlier, we have wonderful drugs against malaria. We also have very good vector elimination strategies, but the challenges that we are facing are multifaceted. So for example, the malaria causing vectors are beginning to spread through different terrains at the moment. So there is a huge change driven by the climate change aspects that the malaria spreading vectors, the mosquitoes, are actually inhabiting in new areas that were previously not prone to malaria, right? And there are multiple forms of asymptomatic infections and relapse happening, including the zoonotic parasites. There is a major problem with malaria, which is it's a disease affecting the poor people. So in the marginalized population, malaria elimination is not necessarily a top priority. So, and the problem is the mosquito nets are a wonderful way to prevent mosquito bites, right? But as you can see from the image at the bottom, these mosquito nets that were insecticide-treated mosquito nets were used for fishing rather than preventing mosquito bites. Similarly, there are a number of initiatives where the drugs are not formulated properly, or the drug usage is not controlled properly, thereby again increasing the possibility of drug resistance. So all of these will probably give you an idea about how challenging is the problem of malaria elimination in the world. Okay. Sorry.
You carry
Please
on.
continue.
Okay. All right. Okay. I thought somebody is trying to tell me something. Okay.
No.
So the malaria parasite life cycle, as you have seen in your college textbooks, is very complex. So the parasite has multiple life stages alternating between the mosquito, the liver of the infected people, And eventually from the liver, they develop into hepatic merozoites. And these merozoites will come out into the blood circulation. They infect the RBC, and then they go through very specific deformation inside the red blood cells. So on the right side is a schematic representation of the development of the malaria parasite inside the red cell. So this is an asexual replicative phase where the parasite will extensively degrade hemoglobin for the metabolic activities. And almost all of the malaria associated clinical pathological situation comes from the red cell infection stage. So the parasite called merozoites, which are shown here, actually get into the red cell. They invade the red cell, and upon invading the red cell, they form the yellow-colored vacuole called parasitophorous vacuole. And within the vacuole, they develop into a parasite stage called a ring stage infection. And from ring stage, they go on to develop into trophozoite, which is when the hemoglobin degradation happens primarily. And then over the next couple of hours, they multiply into a number of merozoites, as you can see at the bottom image. And these merozoites are invasive. And so the total life cycle takes about 48 hours. At the end of the 48th hour, these merozoites break open the red cell, they come out of the red cell, and then they can start another cycle of infection. So the interesting thing is, all that egress followed by the invasion happens within about 2 minutes. And if you can kind of slow down the egress or invasion process, you can basically avoid a next cycle of infection, which is a very interesting therapeutic window. So my lab is quite interested in that therapeutic window, like I mentioned, because the parasites are maturing inside the red cell. They have to break open the red cell membrane and the vacuole membrane and come out. So it involves biochemical and biophysical factors, right? And it occurs in a very rapid manner. So the entire process of parasite release and invasion happens within about 90 seconds. And this process is very highly regulated. So there are specific proteins that are engaged in unique steps in that egress and invasion process. And the red cell itself, the permeability of the red cell is compromised towards the late stage of parasite development. And the extracellular parasite that just came out of a red cell looking for the next red cell, they can only survive that external environment for a short period of time. So if you have an anti-invasive molecule or even an antibody that is able to bind to that merozoite, then you are preventing the next cycle of infection. So in short, the egress process followed by the invasion happens in a very short period of time. There are multiple stages in that 90-second window, 90-second to 120-second window that you can target using either small molecules or antibodies. But the interesting thing is we do not know a lot of mechanical details about the egress and invasion pathways, which is what we are trying to understand in the laboratory. Okay, and today's talk will focus on the red cell tropism, which actually means that many of these malaria parasites, particularly Plasmodium vivax, which is the most common malaria parasite in Asia, and many of the other parasites, Plasmodium cynomolgi, for example, which is highlighted in red, these parasites have a very specific affinity towards immature red blood cells. So these immature red blood cells are known as reticulocytes, and parasites such as Plasmodium vivax, they can only infect these immature red blood cells called the reticulocyte. And we do not actually know what make them preferentially invade the reticulocyte and what kind of molecular partners are involved in that recognition and invasion pathway, right? So which is actually forming the fundamental principle behind my talk today. And I have already mentioned this. On the figure on the left side is showing Plasmodium vivax invasion of the blood cells. So I would like you to focus on the three categories of the host cells, which are mentioned at the bottom. Normocyte meaning mature RBC. So those are your biconcave shaped mature RBC that is in your circulation. And then you have two other population. One is thiazole orange low, CD71 negative red cells. So these are kind of intermediate cell. And the green one is your reticulocyte. So these cells contain residual RNA. They also contain some of the surface proteins including transferrin receptor. So CD71 is called the transferrin receptor, which is actually an important protein internalizing transferrin protein for developing hemoglobin within the red cell. So the reticulocytes, which are highlighted in the green color, is still containing RNA and residual DNA and some membranous organelles, while the red one is an intermediate form as they become the dark colored thiazole orange negative, CD71 negative mature RBC, which we call the normocyte. And Plasmodium vivax in the laboratory as well as in the human being, they can only infect this immature form of the red cell, the thiazole orange positive, CD71 positive reticulocytes. And we have done similar experiments in the laboratory, also using Plasmodium falciparum, which is the lethal variety of the parasite. And even these parasites, Plasmodium falciparum, they also like to infect reticulocytes, but they can infect all stages of the red cell. Yeah, so that forms the fundamental of the tropism of the malaria parasite towards the reticulocytes. And why is it important? Because the erythrocytic stages, like I mentioned at the beginning, they present the most abundant parasitemia. So this is when you have the highest number of parasites in the circulation. They're also responsible for the symptoms of the malaria as well as the transmission. And there is a lot of evidence showing that RBC tropism is important for the zoonotic germ. So I'll tell you as an example that Plasmodium cynomolgi, and the cynomolgi is a monkey malaria parasite, and Plasmodium cynomolgi in the monkey, they can infect any red cell, any stage of the red cell. But Plasmodium cynomolgi, when you kind of adapt in the laboratory, into human blood, in the human blood, they will only infect the reticulocytes. Yeah, so the reticulocyte affinity has something to do with the zoonotic germs, which is something very interesting and very important to look at. Now, the question is, how are these parasites identifying a particular stage of the red cell through membrane receptors, maybe alternating between the cytoskeletal arrangement of the red cell, because for the parasite to get into the red cell, it has to make localized cytoskeletal proteolytic events, whether one cell type is different in terms of the cytoskeletal arrangement, or it is just a metabolic arrangement. So in the reticulocyte, they still have RNA, I mentioned earlier. They also contain a TCA cycle and intermediates. So for an intracellular pathogen, a reticulocyte is much more robust and much more rich in terms of the metabolic availability, right? So maybe the parasite is taking that as an opportunity to go into the reticulocyte rather than a mature RBC that doesn't have as many resources. So to address that problem, first we have to define the reticulocyte. So I kind of showed an image of reticulocyte infection of Plasmodium vivax earlier. But if you look at the maturation step of the reticulocyte, starting from right to left here, the CD71 high reticulocytes are kind of large cells. They stain very abundantly for the reticulocyte, as indicated by thiazole orange. And over the next 20 to 30 hours, they become CD71 negative biconcave shape red cells called the normocyte, which they don't contain RNA and they don't contain CD71, which is a membrane protein of the red cell, right? And this process happens in about 20 to 30 hours. And the interesting thing here is that if you are taking a peripheral blood sample for your experiment, they only contain about 2% reticulocytes. But an alternative would be to go and collect umbilical cord blood, which contain about 6 to 8% reticulocyte. So definitely, umbilical cord is a better source for the reticulocytes. So there are a number of challenges, right? So for example, low abundance of the reticulocyte. We wanted to go to umbilical cord because it had about 4 times larger number of reticulocytes. And in Singapore, when we started this work, KK Hospital, which is a neonatal clinic, which is about 10 kilometers away from our university. So we developed a collaboration with the KK Hospital, from where we were getting umbilical cord blood every week. And until that time, there was no specific marker for young reticulocytes. So we decided that we will take CD71 which is the transferrin receptor protein as a marker for immature reticulocytes. And then we had to develop a method to purify the reticulocytes. So there are different ways of doing it, including density gradients using Percol, but the robustness of that assay is not great. So you can only get about 30% pure population. So we wanted to develop a better way of purifying reticulocytes, where we started doing a magnetic sorting. So we developed a very nice antibody against CD71, conjugated that with magnetic microbeads, and then you can incubate your cord blood with the CD71 antibody conjugated with the beads, and then purify the reticulocytes using a magnetic field. So this is essentially what we did. So you take your blood cells, mix them with CD71 conjugated nanobeads, incubate it for half an hour, and then purify them on a magnetic column. So as you can see, any reticulocyte that is bound to the magnetic, the nanoparticle will be able to segregate in the magnetic column. And then you take it out and then purify, you can get nearly homogeneous purification of reticulocytes. So if you look at the stream, at the very bottom, you can see two types of red cells. One is called CD71, where you can see these blue color markings. Those blue color markings are indicative of reticular matter, which is the RNA protein complexes, which is a characteristic feature of the reticulocyte. And on the right side, you can see the mature RBC. So all of a sudden, using the cord blood, umbilical cord blood, and magnetic purification system using anti-CD71 antibody, we have a very nice way of purifying the reticulocytes. And this image kind of highlights that you can do a subvital staining. So this is a stain using new methylene blue on the left. You can see that particular staining indicative of the reticulocyte, and on the which also has a very different morphology. So if you look at the DIC image, you can actually see that the reticulocytes are actually not biconcave in shape, and compared to the mature RBC on the right side. And we wanted to also do some quality control. So for example, we wanted to ensure that these the red cells that we are purifying are actually abundant for CD71. So we did fluorescence microscopy for the reticulocyte and normocyte using alpha CD71 antibody. And we were able to see that, okay, these reticulocytes, which are pure populations, were having CD71, while whatever was the flow through, which is CD71 negative, in other words, mature RBC, normocyte, they didn't contain CD71. And these experiments were again validated using Western blot experiments. So one of the very interesting things we wanted to look at is how are these cells different? You can purify immature and mature RBC, but what are the major differences between these two cell populations? One of the things we always knew was that there is change in the mechanical property between these two red cells. And we had this wonderful technique available with the lab, where you can actually do a micropipette aspiration of the membrane compartment. So what happens here is that, like the micropipette that we use in the lab, so you actually have an aspiration pressure from behind. You bring the micropipette close to the red cell, and you apply a very specific amount of pressure from the suction pressure from the back end. And the membrane of the red cell will go into the micropipette. And how much of the membrane gets sucked into the micropipette will tell you about the membrane rigidity of the red cell, right? And with that, then you can measure the membrane deformability indicated by membrane shear modulus. So by doing that experiment, we were able to show that the immature RBC, which is shown on the right side, immature RBCs are much more rigid compared to the biconcave mature RBC, which is shown as CD71 on the left side. So in a nutshell, what the data shows is that immature reticulocyte has a much more rigid membrane. And we wanted to validate that. And to be able to do that, we did a very nice atomic force microscopy imaging. So this is a very new technique that we developed in the lab, where you can take the red cell and then you can do a hypertonic lysis. So what you do is you take the red cell, you put it on a glass slide, and then you do a shear washing using a hypertonic buffer. And through the shear washing, you take out basically almost all of that organic material, leaving behind the cytoskeleton. And in the red cell, the beautiful thing is the cytoskeleton is predominantly made of spectrin and angrin. And from that, you can take the membrane thickness measurement using AFM in tapping mode. And that image you can convert into a skeletonized image. And using artificial intelligence and computational tools, you can kind of measure the area of the mesh, essentially the spectrum length. And that spectrum length measurement indicated that in the reticulocyte, which is CD71 positive, the spectrum length is larger compared to mature RBC. And the larger spectrum length is correlating to the membrane deformability, which we managed to kind of prove using computational tools later. Okay, and the other thing is the area and the size, right? So earlier I mentioned the reticulocytes are not uniform in its shape. The mature RBC, we all know that it is biconcave, but the immature RBC, they are kind of having an irregular shape, I would say. And we actually know that they are slightly larger, but we don't know how much. So to be able to do that, I collaborated with Yong-Kyun Park, who is a professor at KAIST in Korea, and measured the biophysical and phenotypic characteristic of the red cell. So we measured the characteristics such as surface area, total cell volume, and sphericity and things like that. So this data was published last year, which actually showed that the reticulocyte in terms of area is about 30% larger. And same with the cell volume. It has about 30-35% larger cell volume. Essentially, a reticulocyte is a larger cell with a lot more area. And if you think about infectivity, the parasite, the malaria parasite, for example, probably be able to identify a cell that is larger, right? But that is probably not all about it. There may be other biochemical factors which we need to identify. And that is precisely what we try to do. So it's a larger cell, point taken. Maybe it is easier for the red cell to get in. It has also a rigid membrane, which I showed the data from AFM. So the rigid membrane, in terms of physics, it is easier to break through a proteolytic cleavage because the tension is larger. And for a membrane that it has a larger, higher tension, it may be easier to poke a hole when the parasite want to get in. But that's probably not all about it. There may be other components, but there may be other protein components which we don't know about, which we wanted to find out. So to be able to do that, we wanted to do a mass spectrometry-based proteomics analysis. Now, the challenge with the red cell is that the red cell has a lot of proteins, I think close to 2,000 proteins, but the majority of protein is hemoglobin and spectrin and ankyrin, like I introduced earlier, and some of the other proteins such as band-3 anion exchange protein. So to be able to get a good proteome coverage, you have to decrease the proteome complexity. So because you don't want to identify a lot of hemoglobin peptides in your mass spec, right? So to do that, we took the red cell, we took the reticulocyte, we did a saponin-based lysate, and then fractionated that protein into membrane and soluble protein. So in the membrane, you look for membrane proteins. In the soluble, you look for metabolic components, metabolic enzymes, and things like that. And you can see on the Kumasi gel on the bottom left, you can see a very nice separation of membrane and soluble. In the soluble, you can see a band below 10 kilodalton, around 10 kilodalton. That is your globin band coming from hemoglobin. While in the membrane fraction, you can see the high molecular weight protein, which is indicating of spectrin alpha and beta, and band 3 and so on and so forth. And using that sample, we did mass spectrometry and identified about 2,000 proteins in the reticulocyte. Now, we were particularly interested in identifying proteins which are abundant on the reticulocyte, possibly to look for the proteins which are more present in larger quantities in the reticulocyte that may be acting as a receptor for the plasmodium infection. So for that, we looked through all the 2,000 proteins, and specifically we looked at proteins which are abundant on the reticulocyte in the membrane fraction, potentially glycoproteins, and trypsin sensitive proteins. Why trypsin sensitive? Because we know that if you take a reticulocyte and you do a trypsin treatment, and then these reticulocytes can no longer be invaded by Plasmodium vivax. So if you are looking at a receptor protein on the reticulocyte that is able to be recognized by Plasmodium vivax, it cannot be, it has to be a trypsin sensitive protein. Yeah. So using that criteria, from 2,000 proteins, we looked at a bunch of very interesting proteins. majority of them, as you can see in the receptor profile, CD71, which is the marker we used for enriching the reticulocyte, was about, this is log 2, so you have to multiply the number by 2. CD71 was about 8 times more abundant in the reticulocyte. A number of other proteins which are indicated as known plasmodium receptors, which are glycophorin, BAFF gene, DAR, complement receptor, there was not much of a difference in abundance between mature and immature RBC. So this is basically an abundance profile. So trying to understand which proteins are present in more abundance in the reticulocyte. Yeah. And interestingly, we found 3 other proteins. One is SMIM1, second one is CD98 and ephrin. These 3 proteins were primarily only present in the reticulocyte and almost lost during the maturation. Now the question is, because they are on the membrane, because they are glycoproteins, because they are trypsin sensitive, and because they are mostly present on the reticulocyte, are they receptor proteins recognized by the Plasmodium vivax? To be able to do that, before doing that, it was a very challenging project because these proteins were not really being explored by other people. So they are relatively newly identified proteins, and there are no reagents available. So you don't have good antibodies, you don't have peptides. So we had to start from the scratch, developing our own antibodies and peptides against SMIM1, CD98 and ephrin, which we did. And we also validated our mass spectrometry data using the antibodies in immunofluorescence, clearly showing that these proteins, SMIM1, CD98, ephrin B1, all of them are primarily present in the reticulocyte and kind of lost during the maturation. So what is next? So at the same time, there was a publication that came in 2016 from Martin Olsen and Jill Story's lab in Sweden, where they actually showed that this protein called SMIM1 is the well blood group antigen. So if you are interested in hematology, you can read up, but what it shows is that there is a well blood group, and there has been incidences where the well positive blood was given to a well negative individual during transfusion, and there was hematological cross reaction and leading to people dying and all of that. So Martin Olsen's lab actually showed that this protein called SMIM1 is the well group antigen. And so we collaborated with them. They gave us recombinant peptides and antibodies for SMIM1. And with that, we went to the field. So this is an interesting phase of our research where Plasmodium vivax, you cannot grow them in the lab. So you have to go to the field where there is Plasmodium infections. And luckily for us, in the Thailand, Myanmar, Cambodia border, there is a place called Maysot, where Oxford University has a field station studying on malaria. And we collaborate with them. So you go to the field station during July-August period, there is a lot of vivax infection. You get these samples and then you can do your experiments and come back to Singapore. So that's exactly what we did. We flew to Thailand and did the field experiments using anti-SMIM1 antibody and peptide, very clearly showing that the recombinant SMIM1 peptide was able to block invasion of Plasmodium vivax into the reticulocyte. Similarly, we were also able to show similar data in Plasmodium cynomolgi in the laboratory, which is a parasite very similar to vivax infecting the reticulocyte. And the other protein was CD98. So that is another very abundant protein on the reticulocyte, a very large protein, and it is an amino acid transporter. It's a multi-pass membrane protein on the reticulocyte membrane, important for transporting certain amino acids into the reticulocyte while they are still metabolically active. Yeah. So this is a collaborative project between Bruce Russell at University of Otago, Laurent Renier at A-Star Singapore, and my lab. So we developed antibodies and peptides for CD98, and we showed that these antibodies are actually quite good. And then once again, we went to the field in Thailand, tested these antibodies, monoclonal and polyclonal antibodies against Plasmodium vivax isolates. And very clearly, we were able to see that if you are adding anti-CD8 antibody, the parasite invasion is blocked. And on the right side, showing some GIMS, they don't look very, very appealing, but this is actually what you can get from the field isolates, and showing that Plasmodium vivax invasion is abrogated if you treat the culture with anti-CD98 antibody. This is a paper that we published last 2 years ago in Nature Microbiology. And now the most interesting thing is, it's good to show that Plasmodium vivax is utilizing CD98 as a receptor to get into the reticulocyte. But that is not sufficient. So you have to kind of identify what in the parasite is able to recognize CD98. And for that, we continued working on it together with Lauren Ray and Bruce Russell. We developed a peptide library, protein library, basically about expressing 42 different Plasmodium vivax invasion receptors. And then we developed an assay where you have these different mammalian cells expressing one or the other Plasmodium vivax invasion proteins. And then you stain the reticulocyte with a green fluorescent protein and put it together. And for each of the library, if they are binding to a particular Plasmodium vivax because of expressing a vivax receptor, then I'm sorry, I think I told it wrongly. So we have these mammalian cells expressing Plasmodium vivax receptor libraries, one in each of these conditions, and then you take the reticulocyte, pre-stain it with the green dye, and then you add. So essentially, if these mammalian cells are binding to the reticulocyte, then you get green fluorescence. So from that assay, we were able to see that this particular protein called PVRBP2A, so those mammalian cells expressing PVRBP2A was able to bind to the reticulocyte, showing that CD98 is probably being recognized by RBP2A. And then we did co-immunoprecipitation experiments and things like that to actually show that these two proteins are interacting. Yeah? We again validated these experiments using surface plasmon resonance and so on and so forth. Yeah, so this data I showed about SOMIM1. Sorry, I think I jumbled one of the slide. And SOMIM1, again, this is a very challenging protein. So one of the challenge with the red cell is that, the red cells are translationally inactive. So you cannot do much genetic manipulation on the red cell. But we worked with Manoj Durai Singh's lab at Harvard, where they have a very nice erythroleukemic cell line system where you can still do genetic manipulation. So these are called the JK1 cell lines. So you can do knockdowns of the red cell proteins and in the erythroleukemic cell line, and then you can use it for malaria infection experiment. So essentially what we did was we used these JK1 cell lines, knocked down the expression of two proteins. One is BEL, which is the SOMIM1 protein, and one is basagine, which is a well-known Plasmodium falciparum invasion receptor. And with these knockdown experiments, then we started infecting them with falciparum or vivax. So this experiment showed us that falciparum invasion is not affected if you knock down BEL expression, which means falciparum does not require SOMIM1 for invasion. But vivax invasion into the reticulocyte was significantly reduced if you don't have BEL antigen expression, which is again indicating that BEL antigen is important for vivax invasion and not for falciparum invasion. So these are the latest results that I showed. But the question is, why are these parasites interested in a particular host? Whether it is because of a metabolic advantage, whether it is protection from damage, and that actually means that the reticulocytes are having a different architecture of its membrane, and they are not so much in the peripheral blood. The reticulocytes are primarily in the bone marrow, right? And in the bone marrow, if these parasites are infecting, they would be better protected from a lot of damaging agents, including drugs probably, right? So we have actually data, which I'm not talking about today, that the parasites that are going into the reticulocytes, I'm talking about falciparum, they are better protected from even drugs like artemisinin. So essentially, what this data showing is that there are membrane receptors which are differentially present on the reticulocyte, acting as a recognizing marker for Plasmodium vivax to get into them. There is a different assembly of cytoskeleton and cytoskeletal proteins that possibly the parasite is manipulating differently to get into the red cell. And possibly there are also metabolic reserves in the reticulocyte that they love so much so that they can multiply better in a nutrient-rich environment. So right now we are trying to develop therapeutic antibodies and peptides targeting BEL and CD98. And we are also trying to look at the role of CD98 and BEL as in the invasion of other malaria species. like I said, the reticulocyte invasion has something to do with clinical drug resistance, and we don't actually know the expression levels of these proteins, well and CD98, among different populations. And all of this is important if you want to look at continuous Vivax culture. I think I mentioned multiple times that all the Vivax experiments, we had to go to the field and collect field samples to be able to do experiments. But if you can find out what make a reticulocyte the preferred host for Vivax, and if we can mimic that condition in the laboratory, maybe we are on the right path to develop Plasmodium Vivax in vitro culture system, which is which would be wonderful. So in addition to the things that I mentioned today, our lab is also broadly interested in drug discovery and understanding blood cell rheology in chronic diseases. So after I came over to RGCB, we are also developing a very comprehensive biosafety level 3 platform where we have humanized mouse models to study various disease conditions. We are also trying to develop an erythropoietic stem cell culture system where you can study red cell maturation associated problems. Single cell mechanobiology, I showed one data showing the membrane deformability measurements, but it's a relatively easy method to look at a number of red cell pathologies, including spherocytosis and so on and so forth. And we also have a strong focus on drug discovery against infectious agents. So we are trying to develop a mass spectrometry based method to look at drug target identification. That is one of the interesting things that our lab is focused on. I look at small molecule inhibitors that can block egress and invasion in malaria parasite. So we had multiple grants on that, and we are trying to kind of, again, look at the thermal melting behavior of proteins when they are bound to a small molecule to identify the drug mode of action. And we, like Simhan mentioned, I am also interested in applying technology into malaria research. We have developed an AI-based model to look at different stages of blood cell development as well as infection. We are also interested in looking at mosquitoes, automatic categorization of mosquitoes, and a robotic system that can do that. So again, red cell deformations, including diabetes, some of these deformations will change the mechanical properties and other phenotypes of the red cell. And we are very much interested in looking at how these different disease conditions, including SARS-CoV-2 infection or immunization, vaccination, all of these are changing the red cell mechanical properties, thereby changing its hemorrhagic behavior, hematological behavior. So with that, I conclude my talk for the day, but not before acknowledging these people. So on the top, you can see my lab in Singapore. And on the bottom is my current lab in RGCB, and all the members of my Singapore lab and India lab. And the bottom, the right hand is our new building, the new laboratory at the Rajiv Gandhi Center for Biotechnology, Trivandrum, under the Department of Biotechnology. So with that, I will take any questions that you may have. Thank you.
Thank you for a hello.
Yes.
It was really a nice talk, and I really enjoyed looking on to the your beautiful pictures, especially the protocols you have developed for cytoskeletals and other protocols. It was really interesting to see antibody tagged with magnetic nanoparticles. It is really nice. As you say that antibody is tagged with nanoparticles and ferritin receptor. So are they going to interact or interfere with each other? Or...
Yeah. So yeah. Very good question. Actually, we know that it is not. So what I'm telling is, so what we have done. So one of the things to remember here is that the reticulocyte is not, not an inert cell. It actually keeps on. So the challenge here is that you have a reticulocyte on top of which you have the protein, and that is bound to the magnetic microbeads, right? And you are wondering whether this will interfere with the invasion. And so what we have noticed is that the antibody microbead interaction is a very, I think in a couple of hours that falls apart because the reticulocyte is maturing and the CD71 is gradually losing. And the reticulocyte is actually always, generating synthesizing for CD71 that goes to the surface. So we don't actually see an interference because of that.
Thank you very much. It is really interesting to see. Thanks.
Dr. Krishna Murari, you have.
Yeah, I'm here. Yeah, it's a really wonderful talk. It's nice combination of field work and hardcore science in the lab, and doing so mass spec with so many proteins and all. It's a Herculean task, I must say that. But so when you were doing the mass spec and you found out 3 or 4 proteins, so I believe there might be more proteins which are responsible for all this, right?
Yeah. So, like I said, we applied a few criteria to be able to look at the most interesting proteins. So we identified more than 2,000 proteins, but we wanted to look at those proteins which may be important in the parasite recognition. So we were primarily focusing on proteins which are on the membrane, glycoproteins, which are trypsin sensitive. So that threw us with these 3 interesting proteins. But like you said, there are a number of other interesting proteins, particularly the metabolic enzymes. So one thing which I didn't talk about today is that there are metabolic enzymes which are part of many pathways, including TCA cycle, which is present in the reticulocyte and not in the normocyte. And now, if you think about a parasite point of angle, this having a nutrient-rich reticulocyte environment, the parasite can basically kind of bypass a lot of its own metabolic activity. Just take that intermediate and load it into its pathway, right? So some of them may be important for long-term survival of the parasite in the reticulocyte. It is something we have to look at. But for now, we will only looked at the membrane recognition part. There is a lot to do.
Yeah, it must be. And I believe to rupture orthocytoskeleton and all those on the RBC, there might be some proteases also involved, do you think?
Yes. So these are mediated by parasite proteases, which are kind of expelled at the time of rupture.
Yeah.
And I have actually shown a couple of years back, it is not only the parasite proteins, but also host proteins. So what the parasite does is they change the calcium dynamics in the infected cell. They kind of recruit a lot of calcium into the extra parasitic compartment. And the red cell contain calcium-dependent enzymes like calpains. So all of a sudden, the parasite is recruiting a lot of calcium into the extra parasitic compartment, activating the red cell calpain. And red cell calpain has a very broad selectivity for substrate. So it can then cleave and the red cell itself, right? So that was the paper Lakshmi Narasimhan was talking about, my first science paper, where we showed the parasite is able to manipulate the red cell, use a red cell protein to cleave the red cell itself. So
it's wonderful.
very interesting domain of work. Yes.
Yeah. So any other question?
Rajesh, at the beginning of the talk, I mean, while introducing you, I said you are a good speaker, right? Because I've heard your several talks before. But I would say today is your best talk that I've ever heard. I mean, this is better than what I've heard before. The flow, the way you would organize the content, and the way even advanced topics, I mean, topics which you identified topics which may be new to people, like for example, micropipette aspirations to measure the deformability of the membrane. You took time to explain that, so even graduate students who are attending it, they'd find it really useful. The talk was well organized, well delivered, and you covered gave a very comprehensive story. At the same time, you introduced us to the diversity of the work you are engaging in. I'm really amazed. Thank you very much. We'll disturb you very frequently. You have so many different projects, so you can give a newer and newer talk every time we invite you. Right? I'm amazed. Thank you.
Yeah.
Oh,
Um,
can I say something?
yes.
Yes, Mackie.
Okay. Doctor, this was really an amazing, and it made me remind my 1995 pass out stages of Plasmodium vivax when we were taught Rosette stage and the Signet stage of Plasmodium. Okay, so this was deeply, and now we came to know that how this because you got involved with science, and now I think you are giving an expression of you are evolved out of science now. So this is a different stage. You are developing very good key markers as well as you're recognizing the how to track it. Malaria is not a small thing. I think 1/3 of the world population is combating with this particular terror. So my question is, this is exactly science, but what is happening outside? What is your perspective about that the approval that got from the USDA and even FDA for the Oxitec-based GM mosquitoes?
Yeah, I believe that.
Because now they are claiming, they are claiming that not only Plasmodium is the one, they are battling for other viruses also like Zika, Chikungunya. So how the story is going to cover in next 10 years? Can you give, because you are deep into it.
Yeah. No, I mean, I read, I'm not an expert in that, but honestly, I think this is where the world is going to be. I think the fundamental biology of the parasite is wonderful, but at the same time, there is a need to have radical changes stemming on technologies, novel technologies that will address the problem in a relatively short period of time, right? So from that perspective, I think the genetically modified mosquitoes is going to be one of the way to move forward. The other thing is, for example, in Singapore, that what we know is the Wolbachia system, right? So they have actually implemented the Wolbachia system to kind of very effectively control the dengue infection, which is still something that is in the pipeline. But of course, there is a lot of things to do. So there is another thing where one of my former colleague who is actually in China. So what they did was that they developed specific fungus, which is naturally infecting the mosquitoes. But at the same time, these fungi were genetically modified to be able to express certain proteins. So the point is, when they infect the mosquitoes, they actually kind of influence the mosquito's ability to withstand or support plasmodium development in the salivary gland, right? And not only killing the mosquitoes, but also kind of terminally avoiding the parasite development within that mosquito. So there are a lot of those angles that are being looked at the moment. And I think in another 5, like you said, I'm more optimistic. I think in another 5 years, people will buy into all of those. And we will have a better chance at eradicating not just malaria, but also other mosquito-borne diseases.
Oh, thank you. Thanks for your updates. Thank you.
Quite impressed.
You are muted, Dr. Munindra.
Okay. Dr. Rajesh, thank you for such a lovely presentation and an excellent piece of work. So I wanted to ask just one question regarding the therapeutics. So your work has identified this well and CD98 proteins, which can be potential therapeutic targets. So if we say use monoclonal antibodies against these targets, so would it also affect the normal working of these reticulocytes?
Yeah.
So how that can be avoided?
Yeah. So this is essentially why we wanted to also identify the partner, the protein on the parasite that is recognizing the host protein, right? So the RBP2A, we have some really interesting antibodies that are coming up now. So it's not about targeting the host protein per se, but maybe the assembly of the host and parasite protein. So CD98, we have a very nice antibody now that can recognize the RBP2A and the protein complex, RBP2A CD98 complex. Similarly, for SMIM1, we don't know what it is binding to yet. So we have to kind of first identify what parasite protein is recognizing the SMIM1 and then take it from there.
Thank you.
Thank you.
So do we have any other question? No, I do not see any other question. So we can.
I think most of the participants are appreciating the talk, as we can see from the messages. Everybody is like the talk a lot.
Thank you.
Yeah,
Thank you. Glad to hear that.
Okay. So.
Okay. Thank you, Rajesh. We'll wind up now, and I'll touch base with you sometime later.
Okay. Wonderful. Thank you.
Yeah. Thank you.
All right. Bye-bye.
Thank you. Thank you. So we join back again at 2:30 for the last talk of our professional development program from Dr. Ravi Dutt Sharma. So do join back at 2:30, and then we also conclude our 5 days program after the talk. Thank you. So should we start, Dr. Ravi?
As you wish, sir.
Okay. Okay, so welcome back. Good afternoon, everyone. Welcome you to the last session of this professional development program. So in this session, we have a talk by Dr. Ravi Dutt Sharma, and he is going to focus on the theme of genomics and transcriptomics. So now I request Dr. Amresh Prakash to kindly introduce our speaker, and then he can begin his talk.
Thank you, Dr. Wali. So once again, good afternoon to everyone. So today we have young, energetic and versatile faculty for the second half talk. So Dr. Ravi Dutt Sharma received his MSc in Bioinformatics in 2006. Then he went to University of Navarra for the completion of his PhD. After that, he enjoyed his postdoc as the KU Leuven, Belgium. Currently, he is the associate professor at the Department of Data Science, MIT Institute of Integrative Sciences and Health. His research area is mainly focused on genomics and transcriptomics. He has developed many algorithms and software using the computational biology and bioinformatics and deep learning. His devotion to his research is laudable. Dr. Ravi has published nearly 30 research articles in the reputed international journals, including the Nature Communications, Neuro-Oncology, Science Advances, Briefing in Bioinformatics, and as well as he has, till today, he has guided the 5 PhD students. So with this brief introduction, I request Dr. Ravi to please come and deliver his talk. Yes, thanks.
So, Dr. Amresh, thank you very much for a nice introduction, though you are sitting left side to me. Let me share my screen. I hope I'm audible, right?
Yes, yes, you are audible.
Okay. So, screen is visible, right?
Yes, screen is visible, everything is fine.
So today, what I'm going to speak in the theme of genomics and transcriptomics. So I prepared to actually showcase or tell you about the software that we have developed in a couple of years, last let's say 5 to 6 years working here in the meeting. And mainly what it does is prediction of transcript structure and then concentration of those alternative splicing based transcripts that can be used for the biomarkers. Okay, so what we call it, the software is PHASE. I will tell you what it is and why we come up with this name. Okay. So just for the shape of all the audience to make understand what it is, I will give you a little bit of background, and then I will come up with what we are doing and how this phase works and what is the philosophy. So most of us, we belong to that biology background, and we know that our body is, or you can say human bodies encompasses of different tissues, and these different tissues are having a distinct cell that we have, and each and every cell actually has lots of different components, but one of them is the chromosome, or you can say the DNA, the ultimate thing, the blueprint of each and every cell. So with this DNA, what comes up is the differential gene expression that make these cells in different tissue work differently and appear differently. So approximately what we have in our human body is nearly 3 billion base pair of this DNA. And these encode for approximately 33,000 genes. Now, these 33,000 genes actually encode for more than 100,000 of proteins. So this complexity in proteome from your genome comes because of a phenomena which is known as alternative splicing. And that's what I'm going to talk about. So this phenomena that we say alternative splicing, where this precursor mRNA process in such a way that it encode for different transcript, but we say isoforms also. And in this case, you can see the introns are removed and these exons are actually combined in such a way that the protein product is going to be different. So just to understand this phenomena, we can take an example. Okay, you can see here that we have 3 exons in one DNA, let's say, and there is intron here. In precursor mRNA, this intron is removed, and these 3 exons are joined together in such a way that it encode for different proteins. You can see here, if the full-length protein comes, then you have a different protein. If you have exon 1 and exon 2, then you have a different protein. Likewise, exon 2 and exon 3 is coming, different protein, and exon 1, exon 3 is coming, in different protein. In some of the cases, this intron may be retained, and this type, this particular transcript will be marked as for the NMD pathway, that is nonsense mediated decay pathway, then there won't be any protein for this. So most of the time, it was thought that it is not a regulation, but it is also a well thought regulation of protein preparation, or you can say production or synthesis. Okay, so depending on this, there are different types of alternative splicing. For example, you can see here a cassette exon, where this exon is gone out, and you have exon 1 and exon 3, and in one transcript, you will have all these 3 particular exons included. So you have 2 types of transcripts here in this case. The second example is for the mutually exclusive. So this green one is having mutual exclusion, or you can say a problem with exon 3. So if exon 3 is included in transcript, exon 2 won't be there. And if exon 2 is included, then exon 3 won't be there. So this kind of alternative splicing is known as mutually exclusive cassette exons. Then we have exon lengthening and also exon shortening type of alternative splicing, which is known as 5' 3' splice sites in exons. Then we also have alternative polyadenylation site type of alternative splicing, as well as we also have alternative polyadenylation sites also, right? So this type of alternative splicing we also found in organisms. And then last type of alternative splicing here, you can say intron retention type of alternative splicing. Okay, so what we did in after coming to MIT University in 2016 and 17, we prepared 2 algorithms to find out cassette exons and intron retention type of alternative splicing using RNA sequencing data. So basically how these 2 algorithms work importantly, or you can say very strongly, and we were able to validate these results very strongly using PCR because of using this junction information. So you can see here, if we have exon 2 as a cassette exon, then you have junction which expand from exon 1 to exon 2. And then there is one junction which is expanding from exon 2 to exon 3. So idea is, if these 3 exons come together in a transcript, then there would be one particular read that actually expand from exon 1 to exon 2. And this is what we say is junction read, and we count those reads. In the same way, we have here exon 2 and exon 3. If exon 3 comes with the exon 2, then there will be some read which expand from the exon 2 to exon 3, and this will be called as junction. We count those reads. In the same way, if exon 2 is gone out, then exon 1 and exon 3 will be joining together, and there will be one read which expands from exon 1 to exon 3. And this is what we say junction 3 here as a junction. And we count those reads. Now, what this read particularly signifies is it is actually shows that exon 1 and exon 3 are expressed together. And that's why exon 2 in the middle has gone out. So this significance of this particular junction is actually used in those in this algorithm. And the same way, what we utilize the information of this junction, these two junction, which is called as flanking junction for this particular exon 2. So with respect to this cassette exon, exon 2, these two are flanking junction, and this junction 3 is called as skipping junction. So what we used here for our analysis, these junction 1, junction 2, exon 2, and junction 3 as the information whether to make sure that whether this particular exon 2 is being a cassette exon or not. And that's it. That's what is, you can see here the expression. So in this case, you can see this exon and these two samples, this one and this one is AL15 and AL5. You can see it is expressing red, right? And the same way junction 1 and this, yes, junction 1 is expressing in this same way and junction 3 is expressing in the same way. But in the opposite way here, you can say this junction is expressed, this one is, I think naming is different here. I should have named differently. This junction 2 should be here. So this is expressing in another condition. So that's what is the skipping junction, what it is doing here, right? So this phenomena we actually try to capture here, and that's how this cassette exon and intron retention in the same way was working perfectly. We were able to target this. Now, we were able to find out alternative splicing in terms of cassette exons and in terms of intron retention, right? And we were quite sure we were able to validate most of the events here. Now, the questions that comes in our mind immediately, that is how many transcripts are expressed in different conditions? What would be the structure of these transcripts if it is expressing? And can we calculate the concentration of these transcripts in each condition? So what we thought, how we can use this spatial information of junctions to find these transcripts and to find out the transcript concentration as well. So what we did is we took a old model that was given, it's called as linear model, and it was given by Lee Wong, published in 2001 for the microarray data, when microarray was on top of the floor, you can say, and there was no RNA sequencing. So what they said that this intensity of the probe is actually proportional. You can say the affinity of the probe and the concentration of these targets plus one error term. What we did is we changed it to mathematical equation, linear algebraic terms. Then it comes like AFGT, where A is the affinity of the probes, F is the feature matrix, G is the property matrix, and T is the concentration, and plus E is the error term again. Now, if we try to use this equation, we need to modify it according to RNA sequencing. And for the RNA sequencing, there is no probe affinity. So there are two terms that actually goes out, and those two terms are, just a second. So these affinity terms and these feature matrix terms go out, and then we have the G. So this G signifies the transcript structure and how many meta features we have, and this T signifies the transcript concentration and the structure of the transcripts. Now, using this equation, what we can do is we can calculate the concentration of transcript given the Y matrix that we have and the transform matrix, you can say, of G. So this G is the structure of we have the gene, how many meta features we have. That's what we can utilize just to calculate the concentration of this transcript. Now, in order to work this with this but small equation, what we need is the accurate prediction of number of transcripts that are expressed in the different conditions and the structures that we have, and then it can work out perfectly. So there has been number of work in this area. Some of the people used number of methods to work out, and one of them was a very important method, that was the non-negative matrix factorization, that is NF in short. So what you do, you have a bigger matrix and you decompose this matrix in two similar matrices, or you can say that can correspond to the structure of this particular gene, and one matrix that can correspond to the concentration, depending on the meta features that you do. Now, well, sometimes it works, sometimes it doesn't work, but why it is not gonna work? So the problem was the not convergence of NMF. Okay, so once you provide a starting point for this NMF algorithm, then it works. Otherwise, it will not work, of course, and that requires actually assumptions. Assumption, how many transcript is going to express, for example, 2, 3, or 4. So if initial assumption of transcript is wrong, then this particular NMF goes wrong, and whatever you will get is the wrong concentration estimation as well as the structure of the transcript. And that's what is the main drawback for this algorithm. So what we did is we made some assumption for our RNA sequencing experiments, and what we said is, if for a single alternative splicing event, there could be 2 transcripts at least. One, if, for example, this exon 2 is a alternative splicing event, then one transcript will have this one included, and another transcript that will have this exon 2 excluded, right? So there would be 2 transcripts, inclusion and exclusion, right? Now, so the complexity of this transcript structure prediction will increase with, if we have more alternative splicing events in a gene that changes with N, the number of meta features, how long the genes you have, number of exons, number of introns, number of junctions, the more bigger the gene, then you have more complexity in this transcript structure prediction, as well as the number of conditions that we have for the experiment. For example, normal of a tumor sample, or maybe peripheral samples, or some more treatment. For example, drug treatment. You have number of drugs that are treated with your samples. So then the number of conditions actually increases the complexity proportionally. So what we did, we proposed a equation that is T equal to N multiplied by D and multiplied by M minus 2. When this M is actually, we should have greater than or equal to 3, at least 3 terms should be there. Either it is intron or exons combination or exon-exon combination. And then we are able to predict the structures. So what we did is now, you can just see one example here. This exon 3 is a cassette exon, and we have different junctions here. And this junction 4 is called as skipping junction for this particular exon 3. And these junction 2 and junction 3 will be the flanking junction because it comes to the exon. So if we take this example for the transcript structure prediction, so what we did, we find out a seed meta feature, and seed meta feature we say is a particular cassette exon, right? And another seed meta feature will be for junction 4 because it is skipping here for the exclusion period. So one transcript will have this exon 3 inclusion, and another transcript will have junction 4 as inclusion. So what we do is we take these seed meta features, both of them in case of exclusion and inclusion, and using a graph theory, what we do is back propagation and forward propagation. How many connections we can have for the exon 4 connecting here, exon 3, exon 4, then exon 5, 6. Likewise, in the back propagation, we go from this particular exon to exon 2 and exon 1. And we try to find out all the possible paths using the graph theory. So using this graph theory, we can find out, of course, the possible path, and down we can have the number of transcripts or the structure of those transcripts that are getting expressed both together. So what we do is, using that, we use a urgency matrix, that is GG, okay, and let's see transcript the transpose of CM matrix, that we say is the connection matrix, or you can say that contains meta features, okay. And once we multiply with this, then we have this GG, and we can create a directed acyclic graph, right? Where this G, this graph is represented, and where B is vertices and E are the edges, right? So in this case, the exons will be our vertices, and edges E contains our junctions, right? That will give the direction of a particular graph, right? Okay, so once we apply this, we know now we have number of transcript coming with the connection that we have here with the junctions and the structures that we have. So for example, here for the inclusion event, where this particular exon 3 is coming here, there are two possible transcripts, right? Where one is here exon 5 and skipping one. So there are two possible transcripts here. In case of exclusion, you have instead of exon 3, there is junction. And likewise, there are two possibilities of having different transcripts. So likewise, in this particular case, we have 4 transcripts that can express, right? So now we know that how many transcripts are there, the number. And we also know the exact structure of these transcripts. So instead of, just to remind you, going back with the assumptions that NMF does, we are directly calculating the number of transcripts and the structure accurately using the RNA sequencing data. So there is no need for doing assumption and no need to use this NMF method. Okay. So once we have this transcript structure accurately predicted, now it is very straightforward to actually put that information in our equation and calculate the concentration. So what we did is we used a data just to try whether our method is working or not. So we used TCGA Barca data, and we did lots of analysis before, and it was published in Cell Death Disease with the Jenny. So this data was already published, so we took this data, and there were lots of differential expressions, nearly 2,134, and we got 6,000 cassette events and nearly 4,000 of IR events, that is intron retention events. So we selected 2 genes for the validation, that was EMI-LIN1, elastin microfiber something gene, which is used for elastic fiber and fibration in different tissues, prostate cancer, for example, and lungs. And another one, the SLK gene, that is a tyrosine kinase, which is important. So we selected these 2 genes. Most of why we selected is we got this cassette event in most in these, I think, all 4 different subtypes as a cassette exon. So there were alternative splicing events selected in both the subtypes. So that's why these 2 genes makes importance, and we also selected for the validation. Okay, so what we did is, first we wanted to be sure whether these particular structures that we have predicted using the phase, whether they exist. So our collaborator, Dr. Jenny, I think Krishna was working with Jenny. What she did is she prepared a forward and reverse primer. You can see with blue arrows here. And using these primers, she was able to validate whether indeed these all 4 transcript structures were expressed in the, I think, cell lines. Right, so here MCF7, BT474 is these are the corresponding cell lines for those subtypes of the breast cancer. And here you can see the predicted concentration for these 4 transcript in different breast cancers. Right. So one major difference that you can see here is the transcript 3. So this transcript 3 is the full length transcript, and for us it gives more or less maximum concentration in all 4 subtypes. And here you can see in the validation it is different. So these 4 figures in the left side are a little bit different from the figures that we got from the concentration estimation from the our phase. Why it is so? That in phase preparation, the normalization what we do is against the library size of the reads. But in this case, the PCR validation what you do here, the concentration is always, or you can say the expression is always taken in the ratio of the full length transcript, right? So that's why it is different here, and it is not directly comparable. But what you can see is the trend here, remaining trend here. So you can see is this red, blue, and this purple one is the getting in decreasing order in the first one, linear way, and in the same way it is here also. The way here it is, you can find out similar over here, likewise here, and also here. The similar trend you can find out that these all transcripts are, of course, leaving the full length transcript. They are expressing in the same way and giving the concentration also in the same way, right? So that's why in the same manner, we validated the SLK genes, right? And we got the similar transcript concentration here. Okay. And you can see here, this one, this blue is lowest transcript here. Here we predicted this as negative. And all of them, they are having the lowest transcript, the concentration. In this particular lumen B, there was no, no concentration for this particular transcript 8. And we predicted as negative. So it also gives one understanding how we can validate them and how this validation works in the wet lab experiment. Okay, so once we have, we are interested, so what is the different in phase from other state of the art? For example, we have slide, cufflink method is there, string tie is there, and it has been nearly 10 to 15 years for the, you can say, in prediction of these structure and concentration. And what is different from these state of the art of the methods? So the main difference, the first difference is in the assumption. So these all methods work in the transcriptome assembly. So first they try to find out the transcriptome of the particular gene or these transcripts, and then what it is going to do is find out the concentration on the basis of those transcript structures, right? So this methodology works for the already annotated transcripts. But what about the novel transcript that you have, where the alternative splicing events are more prevalent, right? So main significant disadvantage is that they cannot find out the novel isoforms, and they always focus on the highly expressed transcripts. And I will tell you how this is a problematic. Okay, so they undermine the low expressed transcripts. So for example, if these 2 transcripts we have in condition 1 and condition 2, and this is a long transcript, you can see here, we have exon 9. And they differ only in exon 5, which has only 3 reads available. right? So once I'm going to make a differential expression of these two transcripts, and because of 3 reads difference in the overall concentration of these two transcripts, it will be not selected as differential expression. And that's why on transcriptome level analysis, these 2 transcripts will be rejected, right? And will not be selected. But in our case, what happens, we first find out this exon 5. And hence, our methodology is not dependent on the transcriptome level expression at all. So we can find out the low expressed transcript also. Okay. Now you can see this graph that has been actually from the same genes, that is EML1 and SIK genes that we have. You can see here the overall concentration here in most of the times for transcript 3, transcript 4, and transcript 7, transcript 8. This overall concentration is more or less same. So these are very prominent transcripts that are differentially expressed, right? But difference in the concentration is not so much prominent, and hence they are not going to be present. And indeed, once you apply the couplings and string tie methods, these transcripts, these both of the genes are not coming up in the analysis. And our phase pipeline actually first detect the alternative splicing, and that's why these are selected here. And we are able to prepare the concentration and the structure. So this is the main difference, how phase work differently from the current state of the art of methods. Okay, now what other things is different here? So coupling can provide differentially expressed transcripts. That's what we discussed. And in our analysis, we find 89 transcripts differentially expressed in HER2 positive data. Okay, and 59 in this ERJ and A. 342 experiment. It is nothing, is the C. elegans experiment. But if we apply phase, in theory, we can have nearly 3,000 transcripts for the HER2 positive data in comparison of 89 time transcript that Cufflink proposed. So because of this phase is based on the alternative splicing units. And in the same way, the 499 in case of this C. elegans experiment, nearly 10 times higher than that. And of course, this could be low expressed or high expressed. If it could be high expressed, then the couplings could have detected them, but that is not the case here. And of course, when we compared, only one gene was overlapping to the results of the couplings. Okay, so another difference in the prediction and in the structure and the concentration here is these slide, string tie, and couplings. They do not provide the selective prediction of the transcript. For example, one is interested in one gene, maybe 2 or maybe 10 genes, but you need to process using slide and string tie and couplings all the whole genome, and thereafter you can do your analysis or focus on that. And it is actually a time consuming. It could be running for 2-3 days and 4 days, and just a wastage of time. But in case of phase, you can apply it selectively, and for 1 or 2 genes, it runs for 30 seconds or 50 seconds, and you have these structures in the end. So this is the big advantage over the other methods. Okay, also slide and string tie don't provide any ranking for the predicted transcripts, but in case of phase pipeline, because ranking is available, you know that which transcripts are higher in the ranking and which are the lower one, and on what particular transcript you need to focus on that. Okay, so now once we compare computationally, so phase uses less resources. You can see we apply our phase pipeline, coupling, slide, and string tie in different 2 data sets. And you can see here that the time taken in minutes, phase has taken only 9.41 minutes for hard to positive data. And in case of cup length, you can see so many hours. Slide, many hours. Okay, likewise string time, so many hours, right? Running 2-3 hours. And phase can does the same thing in 10 minutes only. The peak memory, the phase is using nearly 200. Here, slide is using the maximum, and others are on the lower side. Okay. Other main important thing is the phase. Once the pre-processing is done, everything is stored in the matrices. And these matrices are RNN matrix that has the structure of the gene and the concentration in another matrix that we could say is G count matrix. And these matrices take a few megabytes of data. For example, here you can see the HER2 positive data is 4.3 MB. And for the entire C. elegans data, it is nearly 3.5 MB. But these other methods, they uses direct BAM files, and it is quite problematic to use the BAM files because these are large in data. For example, for a human data, one sample could be 6 GB. And if you have 10 samples, then you have nearly 60 GB. And you can use phase pipeline just for the sake only in 50 MBs. Once the pre-processing is done, you don't need these BAM files anymore. Okay. So now we were interested to calculate the precision recall and F1, which is the standard once you develop any software. So what is the precision, what is the recall, and what is the F1 score overall? And it has been used widely. So we also try to find out the precision, recall, and F1 score for the phase. And you can see here there are I was telling most of the about phase, but here everything is in red zone. And coupling seems to be in the green zone. And we were also surprised. Okay, it is taking less time, less memory, but what is happening here before the precision and recall and the F1 score? It is getting worse, and other softwares are pretty much good. Couplings is giving 0.6. Slide is giving 0.13, and F1 score for phase is nowhere in actually first digit. It is on the second digit. So we were in dilemma whether phase is comparable to these methods or not. So what we did is we tried to find out what is happening with this and recall and F1 score. And that for that, one example should be given. You can see here, this is one snapshot for a gene, and there are so many transcripts for a particular gene reported here, right? Nearly 50 or maybe less than that. Now, all these transcripts that are reported for this particular gene is not expressed in each and every condition. But what is happening here, this precision takes these annotated transcripts, you can see upside the term, and the predicted transcript. So of course, annotated transcript is going to be more always. And since our algorithms is coupling, slide, and string tie, they are dependent on the annotation, right? First, they take the annotation of the transcripts, and then they create the structure, and then they try to find out the transcript concentration. So of course, they are going to find out this transcript, all of them, because they are given in the transcriptome assembly, right? And that's why they have this precision, recall, and F1 score higher than the phase. Now, what to do? Because phase uses novel things. It try to find out the novel transcript structures, novel events. So what we did is we went back and we discovered this novelty index. So instead of that transcript, annotated transcript, we have given this novel transcripts here. In the same way, we change the recall formula for the novel transcript, and that's why we call it novelty index instead of F1 score now, this one, right? So now we have novelty index to calculate, and let's find out how this phase works. And you can see here very precisely. once the novelty index comes, the phase is outperforming all these methods, right? 0.982. And these methods, they are performing well, not as per the like the phase is performing, right? They are also performing fairly well, 0.8, 0.6, 0.7, but phase is performing best out of these methods, 0.98. Okay, and that's why we were sure now that, of course, phase is comparable to the state of the art of methods. Okay, so finally, what we did, we represented this whole pipeline, and you can see here in 3 parts, that is required inputs, pre-processing, and analysis. And phase was submitted by Harsh. Most of the work was done by Harsh on GIT Hub, right? And it was a part of DBT funded project. It was completed, and also this particular pipeline was published recently in the early 2023 in Briefings in Bioinformatics. So what we do here now, we are able to take input from any of the pipeline, okay? Either directly taken the pasta key files from the SRA that is in CVI server, or you have files coming from any experiment, output of any other pipelines. And what we do is take, given the GTF file, we create 2 main matrices, that is read membership matrix that I was talking about, and the gene-wise read counts for the matrices. And once we have these 2 matrices, we don't need all these stuff, the BAM files or pasta key files anymore. So providing the experimental structure as design matrix and comparison for the contrast matrix, what we are able to do with this, we can find out differential express genes, we can find out differential express introns, as well as we can find out differential express junctions. We can also find out differential alternative splicing in terms of junctions, intron retention, and cassette exons. And we can further construct this transcriptive structure, as well as we can quantify the abundance as concentration. We can do, given the clinical data, we can also do the survival analysis. I will tell how this survival analysis is based on this alternative splicing events. So that's what the full pipeline can do, and it is available in GIT Hub. You can check out. There is tutorial for everything, for each and every analysis. Okay, so what extra phase does is the analysis, survival analysis based on the alternative splicing events. So, well, it was already published in Cell Death and Disease. What it does here, the way we were finding our cassette exons by combining the flanking junctions and the skipping junctions, this survival analysis module take into account all these exons and flanking junctions to make sure the survival analysis, right? So there are 2 types of analysis. One is univariate Hooks analysis, and another is multivariate Hooks analysis. And we found that once we combine this particular cassette exon, instead of taking into account differential gene expression, that is up regulation or down regulation, or instead of transcript level of expression for prediction of survival analysis, this exons and combining with junctions is more reliable way of predicting the survival, right? You can just check out this paper, how it is more robust to find the survival analysis based on the alternative splicing events. It's a little bit hazy, maybe I just increase the resolution. Or maybe I stress this particular image, that's right. Okay. So there are a few key points for the phase. You can use to find out the differential expression, alternative splicing, concentration, as well as the structures of transcript, right? In for the real RNA sequencing data, and for predicting the transcriptive structure, it can be applied selectively, gene-wise, right? And this pipeline is developed in R programming language. I think we using the tutorial, anyone would have any bigger problem. For this tutorial is given and everything is provided there. So these are some application that has been already done using the methods. And in fact, this particular method is developed in patches when we required something. For example, cassette exons and intron retention, we wanted to find out more robustly, and we developed that method. Later, we were interested in transcript structure. That's why we developed that method. And next, we wanted to perform the concentration. We developed that method. And likewise, the survival analysis, and then we combined all together. This particular, all the packages, let's say modules together in one particular software, and that's how the phase developed. So you can see here, it is already applied on number of studies, and the robustness is already but reported. For example, here in this ACS Central Science paper, for Nogeni, it is already known. We predicted one intron detention, was validated perfectly, and it is available now. Okay. So this, when we applied phase for the differential gene expression analysis on Mycobacterium tuberculosis, and this is one example where our prediction was validated using PCR, okay, in Mycobacterium tuberculosis in different organisms. Likewise, in rhesus monkey model, right, differential gene expression was validated. It was published in NGChem, and in the end, this is our lab. Little bit small, number of person has been graduated. Nikita graduated yesterday. She defended her PhD, not yesterday, 19th July. Harsh early in this year. Madhavi was in 2020. Kusum is graduating hopefully by next week. So, and this is some of the current and the past member in the lab. And in the end, I want to acknowledge some of them. My collaborator, Arnav, Avinash, Jenny, Jyotsna, Raman, my PhD supervisor, Professor Ankel Rubio Diaz in University of Navarra, mentor, Professor Andrew Andin, JNU, and of course, all the funding agency for the work and support. And I thank you for listening patiently, and I will be happy to answer any question if you can.
Thanks, Dr. Ravi Sharma, for the nice talk. So we will take some questions. So any questions from the audience?
I think I can ask one question.
Yeah,
So I'm not very into this computational thing. So I just wanted to know that if suppose we identify a new gene, so can this phase be used to predict its transcripts also? Means novel gene or novel sequence?
Of course, it can be, but it is based on first alternative splicing. You find out the alternative splicing events, of course, it can be.
Okay.
It requires seed exons or meta features that we give in terms of exon or maybe junction, and then it can predict the structure.
Oh.
Dr. Abhinder, may I also ask a question?
Yeah.
So, actually,
I can see you are speaking, but it's not coming to me.
okay, one minute. Let me
Now
adjust
it
the
is coming.
microphone. Yeah, so I was wondering if there are any limitations actually. Do you think that all the
Limitations are there. Of course, there are limitations.
so
For
which
example,
would be the important ones?
One important one is when the structure is more complex. For example, in humans, we have multiple exons, introns, and junctions.
Hello.
Right? And if multiple alternative splicing events are there, right, 2 or 3 events are there, it takes into account all these 3 events, right? It will try to resolve the structure using those graph theory. And sometimes the structure, the transcript structure, depending on the experiment condition, could be nearly 2,000 or 3,000 or 5,000. And that's what we say is unbelievable, right? This is not possible. These would be so many structure expressed in those conditions. Let's say 3 or 4 conditions. And this is because of complexity of that particular gene. And in that case, we constrained phase that if it goes nearly, let's say more than 500 transcripts, or let's say even more than 200, it is unrealistic.
So
It will
that
not
is
report
something which
them.
is happening actually, or it is
It happens.
being
It happens.
picked up like, okay.
No,
all
it
right.
happens. It happens. So this
I
is one of the constraints where phase will not give you any transcript structure. It means it
see.
is exceeding those criteria.
I see. Thank you. Thank you very much. Great work. Nice to know all these details.
Dr. Rawali, you can open the session for the delegates if they like to ask any question.
Yes, so since we are at the last part of our PDP, the participants can also turn on their cameras and ask if they want to ask any question or they want to give any comments. So are you people able to do it?
So meanwhile,
Anybody
Dr.
who is interested?
So meanwhile, Dr. Ravi, what enrichment you have used for your model?
What?
What enrichment?
Oh, I think we have to give the rights. Dr. Rawali, tell them to give the rights, but they are not opening their cameras.
Maqsood, Mr. Maqsood, if you are hearing, you can give the rights to the participants also to open their cameras.
Yes, sir. Done, sir.
And,
Yes,
and
Amresh,
also
what
allow them
you were
to
asking
speak.
the enrichment, right?
yeah.
Enrichment analysis, we are for the moment we are unable to do that using phase. Well, there are more standard pipeline for, I think, enrichment analysis. For example, David is there.
But we have done, I hope you have done the benchmarking.
Yeah, we did. We try to find out whether our results are significant or not. We are finding relevant pathways. For example, breast cancer data, if we are able to find out the relevant genes for those pathways, which are already well established. We did that. That part I have not included because I thought maybe it's gonna make sleep all the participants.
Oh, so they have some forward selection model and all these things. So what regression model you have applied for your development?
For us, it's not regression model. We have the linear model. We try to find out the linear, you can say, relationship between the concentration, right? The expression, what we say, expression, and with the meta features that we have, and also those, combining those junctions that are flanking and then what are skipping. So that has to
for
be
the transcripts, this survival analysis, they have one very known model also, Cox regression model. Have you checked?
Yes, it is well documented. I have not given more details for that. What we are using, we use there is univariate Cox analysis, and another is multivariate. So in the background, for using these multiple events, junctions, exons for survival analysis, we use multivariate Unicox analysis.
Okay,
so we
thanks.
are not doing anything else there.
Sorry to interrupt you. So I'm actually looking for any question from the audience, because we can also we can talk all these things. So any questions from audience? Many have opened your cameras. Just please unmute yourself and ask question.
You are free to ask the questions. Also, please open your cameras for the group photo.
I can see some
And people can all can feel free to give their feedback regarding the PDP. Any suggestions for all the sessions that were held in the last 5 days? Please
Yes, you can give your feedback live apart from the form which you have to fill.
Very good morning, sir. It was a very nice session and well organized. All of you people made very good effort to organize this FDP and incite us with the conceptually simple and very vast knowledges. Sir, I would like to ask to our speaker, Dr. Ravi Datta, I think. Sir, is there any means the pathway of genetic mutations that causes genetic disorders like single point gene mutations or like enzyme linked genetic disorders? Can it be unmutated to unlock the disease or the linked with the genetic mutations?
But most of the Y work was focused on alternative splicing, right? In this case, I focused on alternative splicing specifically. Of course, there are many things how this alternative splicing and genetic mutation has interpreted. Right? One of my students is working on that to find out whether there is a interplay between genetic mutation and alternative splicing. But I think what you are asking, it is totally different, right? It's not related to that. You want to ask pathways related to mutations or something like that?
Yes, have you ever explored or tried to explore that means acetylation at the purine or pyrimidine or the cytosine bases can mutate the gene expression?
So
And
those comes under epigenetic changes, and of
yes,
course they have a strong correlation.
Yes.
Right. So epigenetic changes, once we talk about, it is not what actually encoded or inbuilt in your let's say genome, right? Like SNPs does. But these are some external factors which comes like epigenetic changes, like acetylation, phosphorylation. There is RDDM pathway for that, right? RNA-dependent DNA
So
methylation.
such epigenetic changes can be cured. Congenital disease can be given.
Now this is a different aspect. Look, I'm not biologist perfectly.
No,
right?
no one is perfect.
No. The question is, I won't be able to answer that thing, right? You need to find out answer specifically for that problem, right? Because my main focus is development of algorithms and other things, not finding the relevance of these. Of course, what we try to find out, it should be relevant to experiment conditions. That's what we do. But that mutations related to whether disease or not, there are number of approaches where you can find out whether a SNP is related to particular disease or not, related to disease or any pathway or not. Right? There are many experiments for that. Right? Even we do this stuff using in silico methodology. For example, MD simulation. We try to find out whether a mutation is deleterious for protein or not, right? Is there any other effect or not? But these are some not specific question. These are some general question you can correlate or not. It depends how you want to do it, right? There are some mutagenesis studies when you do it, right? For finding whether it has any effect further or not. But with epigenetic changes, I don't know any idea. Of course, we try to find out epigenetic changes that is separately. For the moment, we try to find out interplay between mutations as well as alternative splicing, but I have not reached for the epigenetic changes.
Okay, great, sir. Thank you very much.
I hope I answered what you were asking.
Dr. Rawali, you can make a one request to, again, I had already dropped a message to all that they can open up the camera, we can have the maximum photographs, and then we can go for the closure of the session with the vote of thanks.
So anybody who wants to give a feedback, you are free to do. Any comments, feedback?
Sir, I want to, I'm audible?
Yes,
Yes,
yes.
yes, you are to go.
Yes,
Go ahead.
sir. Basically, I'm from Pakistan and I want to say, compliment that from start to end, the whole training was really informative. Thank you so much for organizing such an informative training for all of us. Thank you so much.
Our pleasure.
Thank you.
That you join
It's nice
cross-border
to
also.
have international participants.
Thank you, sir.
Anybody else who wants to say something?
So I think the ice is not broken still in 5 days. Anyway, so we are just dropping the two links regarding the information regarding our MIT Institute of Biotechnology, a hyperlink in the chat box as well as in the our WhatsApp group. Okay, so you can feel free to connect with us anytime regarding anything else or for any collaboration. We are open for that. And second, we are giving you the links of two of our centers. One is the Center for Instrumentation and Research Facilities that is open to all for the on the service basis. There are all the infrastructure which is available for the joint collaborative research and on the payment basis. And third, we are opening up one more that is a MIT Lipidomics Research Center. So all that links you can share with your colleagues. You can keep in your record for the further communication and in contact with us for the future collaborations. So we are really thankful to each and everyone, and this has happened only because of you. And because if we are here, if people have joined and you had made a team, that's why we had reached the day 5. Okay, single can't speak up on this Zoom camera. So it is very important that everybody had given our full support and cooperation and listen with patience. And there may be some errors with us. So we really feel apologetic for that one because of some technical and other issue. And there is some swap off of the programs also. So let me say that to the coordinator of our program, Dr. Maninder Rawali, to please offer the vote of thanks.
Thank you, Professor Machiavelli Singh. So before offering the vote of thanks, I would like to just request you that after we complete this program, we will process your certificates and we will send you within few days. So don't worry about that. So now I would like to give the formal closure and vote of thanks. So on this occasion, as we are here to mark the successful completion of our 5-day professional development program, I express immense gratitude and a profound sense of appreciation. So these past 5 days have been a sort of remarkable journey, and it has all been possible due to the combined efforts, dedication, and enthusiasm of each participant, and also all the faculty members and other participants. So at the outset, I would like to express our deep gratitude to our Honorable Founder President, RBEF, Dr. Ashok K. Chauhan, Honorable Chancellor, Dr. Aseem Chauhan, and Honorable Vice Chancellor, Professor B.B. Sharma, for giving us the opportunity to organize this 5 days professional development program. So an event of this magnitude is not possible without their blessings and guidance. We are also very thankful to Professor Rajendra Prasad, Director AIB AISH, for his continuous support, motivation, and guidance, which have greatly helped us towards the successful organization of this program. I, on behalf of MIT Institute of Biotechnology, MIT Institute of Integrative Sciences and Health, MIT University Haryana, the entire team of PDP, and on my own behalf, extend a very hearty vote of thanks to all the invited speakers and panelists for conducting such thought-provoking and impactful sessions. We have been fortunate to listen to some of the eminent persons like Mr. Gautam Bhattacharya, CEO LSSTC, and also other distinguished speakers from different areas of research in health sciences. So we are grateful to them for kindly accepting our invitation and then gracing the PDP. I would like to thank individually Dr. Himanshu Kumar, Dr. Diksha Tripathi, Dr. Krishna Murari Sinha, Dr. Shipra Chauhan, Dr. Shilpi Minocha, Dr. Rahul Shukla, Dr. Nitin Chitranshi, Dr. Rajesh Chandramohan Das, and Dr. Ravi Datta Sharma for their wonderful talks and giving us insights into the latest developments in their areas of research. To our participants, I extend my warmest appreciation. Your active involvement, enthusiastic engagement, and willingness to learn have been the driving force behind the success of this program. Your commitment to professional development is inspiring, and I have no doubt that the knowledge and skills gained during these 5 days will undoubtedly propel you towards greater success in your respective fields. The program elicited an overwhelming response. You would be delighted to know that we received a huge response from the participants all across the country and also international. As you have seen, we have a Pakistan friend also here. So a large number of students, research scholars, and faculty members, they enrolled and they participated. We had very good attendance on all the 5 days, and I'm thankful to all of you for their patient support and for your patient learning. So I would also like to support all the faculty members of our department, the students, staff members. They have helped us to conduct this program. So we have been fortunate enough to be backed by a team of very motivated and dedicated colleagues who have worked very hard. And it would be unfair for me if I do not specially acknowledge the hard work, planning, and dedication of my colleagues and co-organizers, Professor Machiavelli Singh and Dr. Sarika Chaturvedi. They have been working with me all throughout. And this online session would not have been possible without the technical help of the IT team. So I thank Dr. J.S. Sodi and his team for this. I also like to especially thank the IT help of Mr. Maqsood Ali for his constant support from day one. And he has been an instrumental person in all the online things which we are seeing. I'm also grateful to our student coordinators, Mansi, Naman, Drishti, Rishikesh, for their help in preparing the different reports and other documents. So, as we conclude this 5-day journey of growth and learning, let us carry forward the knowledge and experience which has been gained here to create a positive impact in our workplaces, communities, and beyond. The journey of learning never ends, and I hope we all continue to embrace it with open hearts and minds. Once again, a big thank to you, everyone who has contributed to the success of this professional development program. May we all continue to strive for excellence and work together to create a brighter future. Thank you once again and have a wonderful day ahead. All the best to all of you for your career, for your future. And I think we can meet physically someday. And I welcome you all to visit our campus in Manesar in Gurgaon in person, and so that we can meet offline also, and we can develop more collaborations, more partnerships, and we all can work together for more success. So thank you all.
Just a minute, let me continue with a few missing links. So that is, we like to also pay our thanks to the team. Though there are a lot of people which are visible on camera and sometimes they are not on camera, but they are working on the back end. So first, I thank you for this initiative to Dr. Mahinder Rawali as a coordinator with me and Dr. Sarika Chaturvedi. Along with that, there is a other team of 8 people that you have missed to see on the camera. That were the session coordinators, and along with the session coordinators, they were moderators, but we all are a faculty. These are just designations. Okay. So you can see that special thank to Dr. Manju Sharma, Dr. Sangeeta Kumari, Dr. Anurag Sharma. I think they are all on the camera also. Our Professor Dr. Gargi Bakchi, Dr. Sumishta Das, Dr. Vaibhav Kapuria, Dr. Jini Tomar, Dr. Costa Bandopadhyay, and then yes, Professor Lakshmi Krishna Swami, and then we have Dr. Krishna Murari Sinha, and special thanks to Dr. Amresh and Dr. Nirmal Punitha today to hold this session. So I think with this, we are going to, we can't say we close the session. I think this is opening up of a new network that may carry over forward. Thank you, everyone.
So just a small announcement, the feedback link has been shared in the chat box. So all are requested to fill the feedback one last time. I hope all of you can see it.
Yes, sir.
Yes, sir.
Okay. So please do fill it. And I think I will also post it in the WhatsApp group so that everybody can fill it. So I think we can close the session. Professor Machiavelli, you have to say something more?
Yes, so the group is there. You can share your feedback, and for any excuse regarding the
Hello.
certificate or then our Gmail is there, or on our
Am
website
I
or mail is
audible?
there. We will provide it to you. Yes, ma'am.
Sir, there is one request, the lipidomics links and whatever you shared here in the chat. Could you please share in the WhatsApp group?
Sure, ma'am. Thank you, ma'am,
Yeah,
for
thank you,
sure,
sir.
inviting us. We will share in that group also.
Yeah,
Sure.
yeah. Thank you, sir. Thank you very much.
Definitely. Welcome. So we will keep on posting our other feedbacks and the updates regarding our upcoming programs. So be in touch. Thank you. Sincere gratitude to each and everyone. Please. Thank you.
Thank you. Thank you all.
Thank you, sir.
Thank you.
Thank you, sir.
I think Dr. Ravali, you can share it in the WhatsApp group also. Share the links of CIRF and ALRF in the WhatsApp group. You are mute. Dr. Ravali, you are mute.
We can close the session.
Yes, we can close the session.
Okay, Maksud, thank you. You can close the session.
Thank you, Dr. Masood, sorry, Masood ji, and to the team of Professor J.S. Sodhi for all the technical support.